Let's go ahead and talk about precipitation and particle agglutination methods in serology. So precipitation and agglutination are unlabeled immunoassays. These assay methods produce a visible expression of the aggregation of antigens and antibodies through the formation of a framework in which antigen particles or molecules alternate with antibody molecules. Precipitation is the term for the aggregation of soluble test antigens. Precipitation is the combination of soluble antigen and soluble antibody to produce a visible insoluble complex. Agglutination, on the other hand, is the process whereby specific antigens, such as red blood cells, aggregate to form larger visible clumps with the corresponding specific antibody that is present in the serum. Okay, the IgM class of antibodies is the best agglutination and precipitation antibody. Furthermore, IgM classes are almost exclusively used as reagents, and this class of antibodies is the one that is most commonly detected in patients as an analyte, for example, rheumatoid factor. Agglutination of particles to which soluble antigen has been absorbed is a serum method of demonstrating precipitants. Examples of artificial carriers include latex particles, charcoal, and cells. In latex agglutination procedures, antibody molecules can be bound to the surface of latex beads. If an antigen is present in a test specimen, the antigen will bind to the combining sites of the antibody exposed on the surface of the latex bead, forming visible cross-linked aggregates of latex beads and antigen. Agglutination is when you have antibodies clumped with antigens that are naturally occurring on an organism. That is called a direct method, or they clump with antigens artificially placed on latex or charcoal particles, and that's called an indirect or passive method. In hemagglutination, we use something that is cellular in nature, okay? Whereas in agglutination, we use a particulate in nature. But in hemagglutination, we use something that is cellular in nature. We detect antibodies to antigens that are naturally occurring on red blood cells. That's called direct hemagglutination. One example of that is the heterophile test for infectious mononucleosis. We also have indirect or passive hemagglutination, where the antibodies react with antigens on blood cells that have artificially been placed there. Agglutination and hemagglutination occurs in two steps, and those steps are called sensitization and lattice formation. The first step, sensitization, is the physical attachment of antibody to antigen. It's when an antibody physically binds to a cell or particulate, thus sensitizing it. In lattice formation, tiny particles from chemical bonds begin to form much larger bound particles until the potential to become one solid particle through cross-linking starts to occur. For this to happen, there has to be an appropriate amount of antigen-antibody concentration. The antibody size is a consideration that we have to think about. IgM binds better because it is larger, and the number of combining sites of antibody and antigen sites on a cell or particulate has to be considered. In IgG, there's only 2 combining sites, and in IgM, because it's a pentamer, there's 10 combining sites. Hemolysis and neutralization is when a red blood cell is used in an indicator system to show that an antigen-antibody reaction has occurred. The patient's serum containing antibody and added to a specific antigen will bind. Red cells from a non-patient source is added to serial dilutions of patient samples. Red cells at the endpoint that do not lyse are neutralized by the antibody and bound up and will not succumb to lysing. So this is another serological method, hemolysis and neutralization. Here, patient serum contains antibody and will bind to antigen. RBCs from another source is added, and since antibodies are bound, they will not lyse red blood cells. We've already talked about some of these labeling techniques, but I just wanted to recap. I wanted to talk a little bit about direct fluorescent antibody testing and indirect fluorescent antibody testing. In direct fluorescent antibody testing, the antigen-antibody reactions are seen microscopically with a conjugated antibody. The fluorochrome is fluorescein isothiocyanate, or FITC, and the patient patient directly collected samples provides the antigen source, and that would be things like nasopharyngeal cells or columnar epithelial cells or endocervical cells, like if we're looking for chlamydia. Okay, the antibody in the kit is tagged with fluorescein isothiocyanate, and if the patient is positive, the specimen is going to fluoresce using a fluorescent microscope. Some examples of direct fluorescent antibody testing would be things for RSV, herpes virus, chlamydia, and CMV, also gonorrhea. In immunofluorescence testing that is indirect fluorescent antibody testing, antibodies in patients' serum are going to link up to a fluorochrome and a donor cell or parts of a donor cell that is rich in antigen. The cells are donor cells like human epithelial carcinoma cells. For example, a person with lupus makes anti-double-stranded DNA antibody. This patient's serum will fluoresce with the rich and plentiful source of double-stranded DNA found in the protozoan known as Cryptidia organism. Enzyme-linked immunosorbent assay is also known as EIA, and it's designed to detect antigens or antibodies by producing an enzyme-triggered color change. It uses technology that provides an objective measurement of light, and that would be optical density, and provides a numerical result. It's generally used to measure total antibody in fluids, either IgM or IgG antibody, depending on if we're looking for an acute disease or immunity. This method evaluates the catalytic properties of enzymes to detect and quantitate immunologic reactions. This enzyme-labeled antibody or enzyme-labeled conjugate is therefore used. The enzyme with its substrate detects the presence and quantity of antigen or antibody in the patient specimen. Again, there are three types of indirect immunoassays used to detect antibody in patient serum. We've got non-competitive immunoassay, competitive enzyme immunoassay, and capture enzyme immunoassay, which is also known as sandwich ELISA. These are used in bacterial or viral antibody tests to determine if our patient has the antigen for the bacteria or virus, or if they are making antibody in response to coming in contact with an antigen of a bacteria or virus. A precipitation reaction involves the diffusion of soluble antigen and antibody. Precipitation reactions can be measured by light refraction or by using a nephelometer. Precipitation reactions in gel are not as commonly performed in the clinical laboratory today. Immune precipitation methods in gel can be classified as passive methods or those using electrophoresis. Immunodiffusion is used for the quantitative study of antibodies or the identification of antibodies or proteins in serum. One method is called radial immunodiffusion, or RID. Here we have a protein substance or antigen that is placed in a gel media with corresponding antibody. As the antigen starts to diffuse from the well, the antigen-antibody combination starts to occur. When equivalence is reached between antigen and antibody, a lattice formation is now stable and a visible precipitation ring can be observed. The ring can be measured to determine the amount of antigen in patient serum. And if you take a look at that image on the right, basically what we've done here is we've added 5 microliters of serum into the hole in the gel plate. And then what we see here is a precipitation that forms, and we can measure that ring of precipitation and compare it to a package insert to determine the amount of protein available. We can use RID to look at antibody levels, or we can use it to look at other proteins such as complement or CRP. And those are things that this is a method that we can use to determine the amounts of proteins in patient serum.